Hello students, and in this video I'm going to talk a little bit about prokaryotic cell division and then chromosome structure as it relates to eukaryotes. So prokaryotic cell division is known as binary fission. It is much more simple than eukaryotic division because for the simplicity of the prokaryotic cell compared to the eukaryotic cell. Eukaryotes typically have multiple chromosomes. A lot of the times the chromosomes will come in pairs, and they are linear. So there's a lot more things going on with eukaryotic cells than prokaryotic cells. Prokaryotic cells typically are just made up of a single chromosome. Sometimes they'll have like plasmids or something that are little bits of DNA floating around, but there's usually just one main chromosome that we're focused on dividing, and the chromosome is circular, which makes DNA replication a little bit more simplistic. So start off with a single cell. The cell has a circular chromosome, and you'll have DNA replication start on a single site of that chromosome and go off in both directions. So the origin of replication will continue around until it meets at the other end at the terminus of replication. Once it has reached the terminus, the DNA is divided and the cell is ready to separate the two chromosomes. Now these two chromosomes, as they're being replicated, the cell is also elongating, which you see in this third image here. And then to the point that once the chromosome has been replicated, the bacteria cell is also ready to divide. And so then you have a pinching in the center and a cleavage furrow being formed, and then the separation of two cells, and you have the two daughter cells that originated from the one parent cell. All right, so now we're going to talk about eukaryotic chromosome structure and why this is going to make it a little bit more complex when it comes to the cell cycle for eukaryotes. So when we look at this structure, we can start off with just the simple DNA double helix. So you have two DNA strands going in opposite anti-parallel directions. That is the most basic. This string of DNA molecules then will fold around these different proteins, and these proteins are known as histones. And this is where you get the beads on the string picture when it looks at the chromosomal structure of DNA. The DNA, when it's very loosely packed, will be able to be transcribed and expressed a lot better than it is when it is densely packed. So this is easily read. This DNA structure here, where you start to have folding in on itself in solenoids form, this makes it less active in that the DNA is not actively getting read. Now, during the cell division, you're going to have even further folding until you get a structure that eventually becomes the folded DNA, and typically you see this demarcated as an X structure, and that's what you see during the actual division, although most of the time in the cell the chromosomes aren't nearly this condensed. All right, now I want to talk a little bit about the structure of chromosomes. So eukaryotic chromosomes oftentimes will come in pairs. These pairs are called homologs. So I have two nuclei over here. Both of these are diploid nuclei because they have the pairs of chromosomes. If these dark chromosomes over here on the right and I only had these two light chromosomes over here, they would be known as haploid. But this cell and this cell are both diploid. The difference between these two cells is that this is unreplicated and this is replicated. So over here, the two pairs are called homologs or homologous chromosomes. And so we have one pair of homologs here and another pair down here. Homologs are similar, so they contain the same genes, but those genes can have different alleles. So basically, if you look at the chromosome and you see the sequence of DNA and it's ATCG, another gene is going to have that same ACTG there as well. But it might vary in just a single nucleotide. So instead of ACTG, it might be ACTC on this other chromosome if it has a different allele on it. Now, this chromosome here and this other like chromosome here, these are completely different from one another. So they're non-homologous, they have different genes and completely different alleles because they're different genes as well. So we don't even call them alleles when comparing two different genes from each other. Over here, where we have the replicated chromosomes, each of these chromosomes are actually made up of two DNA molecules. So they're held together at the center at the centromere, and these two identical DNA molecules are known as sister chromatids. They're identical, so they have the same genes and same alleles. All right, I hope that was helpful, and I will see you next time.